All right. Hello, everyone. I hope you're having a fabulous Friday. Welcome to the next matchup for this month's collection battle, where two museum collections are put to the test to see who has the coolest stuff. And I personally guarantee that you will learn something that you did not know before. My name is Chandra Earl, and let's learn about some snails. Our first contender today hails from the Invertebrate Zoology Department of the Florida Museum of Natural History. Working as the Malacology Collections manager, John Slasinski is originally from Virginia, where he earned his master's from the University of Virginia. He currently studies terrestrial snails on islands such as Papua New Guinea, Madagascar, and Bermuda. So if anyone is an expert in beach life, it's John. So how are you feeling today, John? Are you feeling confident? You're going up against Alana.
I ever am.
Our other contestant today is Alana Rivera, the curatorial assistant in the Malacology Collection at the Museum of Comparative Zoology at Harvard University. She did her undergraduate work at Hampshire College, focused on environmental studies in China, and has now been with Malacology for the last 15 years. So Alana, how do you think Harvard's collections can stand up to Florida's collections?
I think we got this.
Well, today we're going to put that to the test and we're going to find out. So for those of you who haven't been here before, this game is played in 3 rounds. For each round, I will give our 2 contestants a theme or a topic. They then have to go into their collections and find the most interesting item that falls under that theme or topic. Something that looks really cool or has a fascinating story. Whichever they think will put them a step above their opponent. At the end, you all will vote who you think won, and to the winner gets the spoils. So let's get started.
What spoils?
My love. Every year on May 1st is May Day, a festival marking the first day of summer. This day is celebrated with food, drinks, dancing, and is also known as Lei Day here in Hawaii, where I am from, or well, I live. Hawaiians set aside this day to celebrate island culture and the culture of Native Hawaiians. And it turns out you can make a lei out of just about anything: flowers, feathers, fruits, leaves, seeds, seaweeds, sticks, nuts, and shells. So for this round, I want our contestants to find me shells that have been made into some sort of shell craft. This can include anything from leis, jewelry, chandeliers, art, whatever you can find that is made up of a shell. And so while they're doing that, let's talk about some Hawaiian leis. Leis are typically given to someone for a variety of reasons or celebrations. Often, the composition of a lei determines its significance. All of the 8 major Hawaiian islands celebrate Lei Day. Each island is symbolized in pageantry by a specific type of lei and a color. The Big Island, or Hawaii, is symbolized by the color red, and its lei is the Ohia lehua, the most common endemic tree in the Hawaiian Islands. This lei is made of Lokelani, the Hawaiian rose, and represents the island of Maui, whose color is therefore pink. The island of Ko'olawe is represented by the color gray or silver, and their leis are made with hina hina, a silvery green shrub endemic to Hawaii. This lei is made of kaunaoa, a parasitic plant that covers other vegetation with its stringy orange-yellow vines. It is the lei of the island of Lanai, whose color is orange. The island of Molokai is symbolized by the kukui nut flower, and its color of no surprise is green. Ilima is Oahu's representative flower, and whose color is obviously yellow. This lei historically was seen as a lei for Hawaiian royalty. The island of Kauai is represented by the color purple, which is the color of the flowers of the mokihana tree. The berries of this tree are used by Hawaiians for lei and were once a favorite perfume. And lastly, the island of Niihau does not have a flower lei, but rather has a shell lei called pupu, which means shell in Hawaiian. Accordingly, the color of this island is white. On that note, let's go see what our collections have for us. So John, do you want to go first? John, you're muted, sir.
Good.
Sorry, yes.
So Rhopophanas are little land snails. They're related very distantly to nerites, which some of you may be familiar with, but this is not what I want to talk about today. I want to talk about kind of a more day-to-day sort of adornment that most of us have. If you all look at your shirts, if you have a button up, unfortunately I wore the wrong shirt today, but you'll see most of them have plastic iridescent buttons, and those are mimicking a mollusk. Those are mimicking the iridescence of unionid clams. So these are freshwater clams. There are 300 species here in North America on where I am, and they've got these gorgeous iridescent interiors, and they tend to be, in some cases, very thick. And when I used to live in Chicago, you could go down any of the major rivers and you would see piles of these unionid clams. And they would have these holes drilled in them. And back before plastic buttons, these animals were fished. There were big fisheries. There was a huge one in Muscatine that employed like 30,000 people. And they would drill out these little blanks. So you would get these button blanks. And then those would be fashioned into really attractive buttons. Like this. And that's why your plastic buttons are iridescent. They're pretending to be these. And after plastic buttons were developed, that fishery really declined in the late 60s. And that was a good thing for the unionid clams. These guys are among the most endangered of all animal groups. And in large part, that's the result of loss of habitat and pollution. So almost all of the rivers in North America have been dammed with many, many dams. And where those dams are built, in the dam, you get a very deep water habitat with no oxygen on the bottom. So any of these animals that used to live in riffle areas with high oxygen and high nutrients were basically smothered. And then downstream of the dam, you often get water releases that scour out all of the sediment. And so there's no place for these little clams to hide, to dig into the mud. This unionid here is only found in the Altamaha River in Georgia. So there's a lot of specificity in where these are found. And in some areas, the entire range of a species might have been dammed and the species may be gone. But these also share some really cool biology. They're parasites of fish. So if you're a freshwater clam, most clams are a broadcast spawner. They send their sperm and eggs out into the water. If you're living in a river, that goes downstream. And that's not really good for a freshwater animal because eventually you'll reach the ocean in most cases. So these have evolved behaviors where many times they will lure a fish. So some of these guys have these little lures that look like minnows that are on the mantle, the body of the clam, and they'll wiggle them around. When the fish goes to attack that, they'll get sprayed with the young of the clam. So instead of broadcast spawning young, female clams hold young, fertilize young on the gills and brood them there until they're ready to parasitize fish. So that's what my adornment is.
That's really cool. Is this the same kind of stuff that's like mother of pearl that's on like guitars?
Yeah, they also, there's some marine species that also have mother of pearl, and those are commonly used as well. So pearl mussels are farmed in Hawaii.
That's really cool. All right, so let's go ahead and turn it over to Alana.
All right. So let me turn this around. All right. So first off, I have to give you a drive-by of one of my favorite hilarious pieces of shell art in our collection. This used to be a desk adornment for a long time for our staff. The cigar actually manages to have like the light be a snail is great. But the actual piece I want to talk about is this lovely handbag, which actually has a catalog number, actually has a label. But these are actually partulas. So these are actually part of the Pacific Island land snails that we're working on. And this particular handbag came to us from the Marianas. And so these have been in our collection since 1947. They're essentially really pretty land snails. You can see the variation. I've done like a rough estimate. There's about 500 snails on this handbag with a lovely Bakelite handle. Yeah.
Do you know when this was made approximately?
Probably in the 40s. It's around when we got it.
Oh, that's really, really cool.
Yeah.
You
So
know,
the same collection that we have, or the same accession that we got at the same time, there's also there's a lei and there's a bracelet as well. But the handbag is sort of the showstopper of the three. When I first started working, I used to say that, like, you know, I needed to borrow this for a night on the town because it's just gorgeous.
It's absolutely gorgeous, but does not look very practical.
No, not at all.
Oh, thanks for sharing, guys. Let's go ahead and move on to our next topic. All right, let me get this situated again. Okay. Go on. Everyone knows that May is for mothers, a celebration honoring motherhood, maternal bonds, and the influence of mothers in society. Every one of us was birthed by someone. All humans are viviparous, meaning that we develop inside of our mother's bodies and we are birthed live. On the contrary, animals that lay eggs that develop outside the bodies are designated as oviparous. There's also a third reproductive strategy, kind of between the two, called ovoviviparity, where the eggs produced are instead kept inside of the female's body, where they eventually hatched and are birthed. Snails, for example, are commonly oviparous, where they lay teeny tiny eggs that hatch into itty bitty snails, shell and all. So for this next round, I want our collections to go find me a species that is not oviparous, but ovoviviparous. So while they're finding their specimens, let me introduce you to Nymphophrynoides occidentalis, a species of frog that is ovoviviparous. This species of frog is one of the only frog species that has this unique ability. So instead of the typical egg to tadpole to frog metamorphosis that occurs outside the frog, that entire process occurs inside the female, all while she's hibernating during the winter. Then when she awakens, she emerges very pregnant, like the female shown here, and births 3 to 5 fully formed toadlets. Unfortunately, these frogs are only known by a couple populations in Africa, where their original habitat has been destroyed due to mining activities. Hopefully they can make a comeback. So now let's turn it over to our collections to see what ovoviviparous animals they have found. Alana, do you want to go first?
Sounds good. All right. So for my choice, I decided to stay with the Pacific Island theme. And so I have here 2 examples of Elasmius luacaense. They're really small. I realize that everything I picked is super small, but I'm going to try and show them to you. So I use my screen. So this gives you an idea. There are some individuals in there. They're super tiny. They're maybe 3 millimeters-ish, super small. But That's true of a lot of these really cool land snails. And that's part of the reason why it makes sense for them to hold their eggs inside of them. Just to give you an idea, because one thing that always sort of bugs me about shell collections is you don't get an idea of the animal. So blown up many times over. And these images are actually from the Bishop Museum. Here's an example of our lovely little critters. They're almost see-through. They're really great. And essentially the advantage of holding the young inside them means that they get to decide when they hatch. It means that they produce many less offspring at one time, which makes them very slow to replace their population. But on the other hand, it means that the one offspring that they have at a time is much more likely to succeed. So it's something like five or so offspring, maybe a year. But that was my choice for this one.
Really cool. They're really, really tiny. I don't know how people find these things out in the field because I would not be able to see them.
You have to get really interested in the backsides of leaves.
That's fantastic. All right, Jojo or John, do you want to go next?
Sure. So I pulled out a couple of things. So like you said, most snails produce eggs. And it's not that many groups that retain the eggs in the female until they've hatched and have much more control over when they are released. And there's a couple of areas on the planet where this is really common. There are montane species that do this. And for that, this is Oreo Helix. This species is Idahoensis from Idaho, of course. And I think it's just a really gorgeous animal. So montane species tend to evolve oviviparity, but so do island species. And that's what I really want to talk about. I want to talk about the family Parchlidae, like the handbag that we saw earlier. And you can see that these were once pretty common species. You know, they had to be very common to be able to produce enough that you could make a handbag out of them, or that somebody could collect this many of this species. But they're not very fast reproducers because they're oviviparous. These will produce only several young at a time. They'll often have a couple of eggs and ready to birth juvenile. And these are really very beautiful snails. But because of the introduction of predators like the rosy wolf snail, flatworms, habitat loss, and other problems, these are becoming very endangered. And many species have already gone extinct or are on the verge of going extinct. And that's one of the downsizes to being oviviparous. You might be able to get to an island and hold your young long enough that they have a better chance of surviving in a completely alien world. But it also means that you're not going to be able to outproduce your predator, out-reproduce your predator. So here's another parchlet that I noticed in our collections a while back. It's a species I described just maybe 5 years ago. And this is the only ground-dwelling parchlet. Well, that's not true. It's unusual in that it's ground-dwelling. And it's even more unusual that it lives in piles of rocks. And this guy also has very few offspring. Usually when you dissect these, they have a single developing egg or embryo. So there's still a lot to know on Pacific Islands, and the fauna is really, really endangered.
It's really interesting. This is why we need these kinds of studies, right? We need to learn more about what these snails do. We need to learn more about their life histories. And that's specifically exactly what our Pillsbury TCN is looking to do. We're looking to digitize and look for all of these species within our variety of collections that we have. Of course, 2 collections that are involved are, of course, MCZ and the Florida Museum. So we're very, very grateful for them we're very grateful for them for coming and joining us here today for this. All right, so let's go ahead and move on to our next topic. Every 3rd Friday of May, which happens to be today, is National Endangered Species Day. This year's theme is Wildlife Without Borders and is the day to learn about this planet's endangered species. Now, these 2 photos are winners in this year's Endangered Species Youth Art Contest. Ironically, the 2 species in these photos, the crested honey creeper and the green sea turtle, can be found in Hawaii. So in accordance of this day, I wanted our collections to find an endangered snail species, which unfortunately is a bit too easy. And while they're doing that, I'm going to talk about the bird in the first art. This bird represented on the winning art entry is an endangered bird found only on the Hawaiian island of Maui. Known in Hawaiian as the akoekoe, the crested honey creeper is most known for its striking white crest and most often feeds on the red flowers of the ohia. Its crest helps pollinate native plants as the bird moves from flower to flower while feeding. The timing of these birds breeding is actually correlated with the availability of ohia flowers. So the birds start breeding as these flowers begin to bloom. Unfortunately, this bird's habitat is ravaged by invasive birds, pigs, rats, and of course humans, so it remains critically endangered. So why don't we go ahead and turn it over to our collections. John, you want to go first on this one?
So the species that I've chosen is probably not the most deserving species, but it is really, really beautiful. This is the Manus Island tree snail. It's found in Manus Island, which is just north of Papua New Guinea, and well, it's part of Papua New Guinea, just north of the island of New Guinea, which is just north of Australia. And because this snail is so beautiful, it was being traded quite a bit and sold. There were markets, there are shell collectors that pick up beautiful shells, mostly marine shells, but in this case, it's a land snail. And conservationists noticed this high rate of commerce and knew this thing was endemic to a single island. And it is one of the very first CITES protected and Fish and Wildlife protected snail species. And that's why I've chosen it. Although many, many, many other Pacific Island land snails are probably in more dire conditions, have populations that are even lower. I think those should also be considered for listing, but I don't think there's much of an appetite for that right now. Anyway, this is the species that I've chosen.
The species is absolutely gorgeous, and that's kind of like the problem. Now that's what happened with the Hawaiian honey creeper too, because of its crest. It became really popular with collectors as well, and so that's what tends to happen to these things, unfortunately. Alana, you want to go ahead and show us what MCZ has to offer for this?
Sure. So I'm going to start off with in the drawer that I'm actually pulling these from, partly because the genera that I'm working with here, Symploea, there's several different species here. So like I'm pulling these, but if you consider the fact that like, you know, they start here and it goes through the drawer, all of them except maybe 2 are extinct. So I could choose any of them and they would be valid choices. And it's the same sort of thing as what John mentioned. Is part of the problem with a lot of these is that, well, they're real small. These ones that I chose, they're not necessarily that attractive. They're like little brown snails. they don't get a lot of investigation necessarily, partly because, you know, they're small and real hard to go find. And so they don't necessarily get updated or listed that often. And so, you know, the nice thing about these, the reason why we have one segregated from the others is they've been imaged, which is actually photos that I've taken of them for digitizing our collection. So it's actually possible to see what they look like a little bit better. I pull up the full image. So, you know, this is an example of what we're looking at. But again, you know, in terms of getting away from what we're actually seeing, you know, you can get an idea from existing species of what the animal actually looks like. And again, they're like just real cute little snails, and they're great. They're just hard to find, hard to define. And there's not necessarily as much interest in going out and, you know, putting them on a list or updating the list to find out whether or not they're still there. So again, you know, like more work to figure out what there is, what we have. A lot of times we're finding diversity in our collections that people don't even realize are separate species, right? So part of it comes from our work that we do in our museums, but also just the sort of general public interest and the idea that these are important and they're important for the environments that they live in.
They're fully grown adults, right?
Yeah, those are full grown, just real small.
Absolutely wild. Because you can imagine like the eggs, you know, for this thing are like fit on the tip of a pencil kind of thing. Super tiny. Ugh. Super difficult to find, but there's a lot of diversity out there if you just, you know, go look for it. Okay. All right, so that go ahead, that ends our 3 rounds. So I've gone ahead and created a poll so you guys can vote to see who you guys think was the most who had the coolest stuff, which collection won this collections battle. So I'm gonna go ahead and let that go and see what people say. I'll let you guys know in a minute. In the meantime, I've gone ahead and posted our emails along with some of the thanks, along with some of the people that we want to thank for this. So of course, you know, we want to thank the Florida Museum and the Museum of Comparative Zoology. And then, of course, the Bishop Museum, where I worked for hosting this, and of course, our Pillsbury TCN for giving us the funding to do so. In the meantime, does anybody have any questions for Alana or John?
Okay.
Alana, those little guys are so cool. How many of those have you photographed?
I'm trying to think. I think I'm up to 230 or so.
Yeah, those are awesome.
Yeah, I think the smallest I've done so far is about a little under 2 millimeters.
That's tough.
Yeah, angling them is not the easiest thing, right? Generally speaking, we have 4 or 5, depending on the shape of the shell, standard images that we're supposed to take. So, you know, turning around something that is real small is real fun.
Fragile too. You got to be real careful with those little guys.
Yeah, don't sneeze.
The barriers inside some of the endodontids, as well as the crocuses, are really cool and probably very hard to photograph.
Yep.
All right, guys. So I've got the polling results here, and the winner today is Alana. So congratulations to MCC. Like I said, the winner gets the spoils. So John, sorry, maybe next time.
All right.
You both did really, really good. I'm so happy you guys could join us today. Thank you so much for this. You know, we all love learning about collections. And join us next month for June. Who is next month? I think it's us versus Oh boy. It's us versus someone. ANSP, I think. But anyway, we'll go ahead and post the details on that. And I will also make this recording available online on our YouTube page in case anybody wants to rewatch or replay. And I will go ahead and post the info about that as well, along with our email addresses and whatnot. Again, thank you guys so much for joining, and we'll see you on the next one. Bye.